There are three types of muscle tissue. Skeletal, it's attached to bones and contracts to move a load. Cardiac, found in the walls of the heart and contracts to create pressure gradients. And smooth, it's elsewhere. This contracts to change tube diameters, make gradients, and expel contents. The three have similarities. All are long cells with an electrically active plasma membrane called sarcolemma. Their sarcoplasm, or cytoplasm, is highly specialized. We also have prefixes which all refer to muscle tissue. But obviously, there are also differences. Location, result of their contraction, neural control, and specialized structures vary between the tissue types. Skeletal and cardiac are similar in many regards, but smooth is extremely different. We see this histology in the laboratory as well. Skeletal, these very long cells are round or cylindrical in shape. They are multinucleate because many stem cells fuse together during embryonic development and the nuclei are kept. Skeletal muscle cells are innervated by somatic motor neurons and therefore under voluntary control. The overlapping myofilaments cause stripes or striations to appear, and we find this tissue in our skeletal muscles. Cardiac. These cells are long but branched. Autonomic innervation implies involuntary control, and the cells are still striated because they contain the same overlapping myofilaments. Cardiac cells contain specialized cell junctions called intercalated discs, and you'll learn more about those in AMP2. And as mentioned, we find this tissue in the walls of the heart. Smooth. These cells are spindle-shaped as they are tapered at the end. The myofilaments in smooth muscle cells are arranged differently, so there are no striations, and we find them in glands, tubular organs, etc. This diagram shows the shapes quite nicely. Some functional differences are listed here. Skeletal, these contract quickly when stimulated, but also tire relatively easily. There is a high degree of control with contraction here, so we will look at graded responses. Cardiac. It's a good thing that cardiac muscle cells are slow to tire. They are arranged in functional groups called bundles, and more about those in AMP2. Smooth. Also slow to tire. These cells are interconnected in large sheets, and there are two types of smooth muscle. We will focus on skeletal muscle, but do know that cardiac cells use the same contraction mechanism and have the same myofilaments. Skeletal muscles are innervated by motor nerves that synapse on the motor end plate. Each organ has one artery and at least one vein for blood supply, and is attached via tendons on the insertions and origins. We also see three connective tissue sheaths. Epimysium surrounds the entire organ. Paramysium surrounds the fascicle, and endomysium surrounds an individual cell. Microscopic anatomy is pictured here. skeletal and cardiac cells are filled with non-membrane organelles called myofibrils. These myofibrils are formed by overlapping myofilament proteins. The overlapping myofilaments form the striations that we see on the diagram here. There are two myofilaments, thick filament because it's fat, and thin filament because it's skinny. Now the very long myofibrils are sectioned off into functional and structural units called sarcomeres. A sarcomere is bound by two Z-discs. We can see the overlapping myofilaments, thin filament and thick filament. Note how the thick filament is sandwiched between thin filaments. Also note how there are areas where there's thin filament only. This pattern causes the striations. The H zone, in the middle of the sarcomere where we see thick filament but no thin filament. The I band, the edges of sarcomeres where we see thin filament. And the A band, the middle of the sarcomere where we see the entire thick filament. Each cell is packed with these myofilaments. Laying on top of the myofilaments is a membrane-bound organelle called the Sarcoplasmic Reticulum, which stores and releases calcium ions. Overlapping the sarcoplasmic reticulum are T-tubules, which are extensions of the plasma membrane, or sarcolemma. A close-up of our thin and thick filaments. The thick filament contains myosin proteins. Note how long tails are packed together in the thick filaments with the heads sticking out. If we let it, myosin will bend its tail and detach its head to a site on the thin filament and pull. The thin filament contains three proteins. Actin. This molecule contains an active site. Myosin heads are chemically attracted to this active site. If we allow it, the myosin heads will bind here. Now, when myosin binds to actin and pulls, this causes the muscle to contract. That means we have to control when this happens. Tropomyosin. This protein sits on the active site on actin and prevents myosin from randomly binding. If tropomyosin is on the active site, the muscle cell will not contract. However, when we want the cell to contract, we will have to make tropomyosin move. Troponin. This protein controls tropomyosin. When it's time to contract, troponin will receive a signal. This signal will cause tropomyosin to move, and myosin's heads will automatically bind to actin's active site. Let's start to look at the contraction mechanism. In order to get a skeletal or cardiac muscle cell to contract, we must stimulate it with a motor neuron. Here we are looking specifically at the neuromuscular junction of a skeletal muscle cell. In other words, the synapse of a motor neuron onto the muscle cell. This is a synapse. So all the neurophysiology of synapses applies here. When the action potential reaches the synaptic bulb, it will open calcium voltage gated channels. Calcium will rush in and trigger exocytosis of the neurotransmitter. That neurotransmitter diffuses across the synaptic cleft to its receptor on the postsynaptic membrane. In this instance, the postsynaptic membrane is the sarcolemma at the motor end plate. If the neurotransmitter binds and we reach summation, we will trigger an action potential on the sarcolemma. That action potential will then travel across the sarcolemma and down those T-tubules. Once the action potential is in the T-tubule, it will trigger calcium release from the sarcoplasmic reticulum. When calcium is released from the sarcoplasmic reticulum, it flows into the sarcoplasm and binds to troponin. This causes a shape change in tropomyosin. Tropomyosin will now slide off the active site on actin. The myosin tail will bend, and the myosin head will bind that active site, forming a cross bridge. Myosin's heads will then pull the actin molecule in a power stroke. This is known as the sliding filament theory of muscle contraction. Let's repeat the sequence. Calcium is released from sarcoplasmic reticulum. Calcium binds troponin. Tropomyosin moves off actin active site. Myosin head binds active site in a cross bridge. Myosin's head pulls the actin in a power stroke. When myosin pulls in the power stroke, each actin will be pulled toward the center of the sarcomere. The two Z discs will be pulled closer together and the sarcomere will shorten. When skeletal muscle cells do this, they generate tension that will move the insertion toward the origin and pull a load. Now, we can't leave myosin stuck here forever. So we will use an ATP molecule to break the cross bridge, and all steps reverse. When a muscle as an organ contracts, these cross bridges will take turns with each other to continue moving and generating a load. As long as action potentials are arriving, this sequence will continue. My point here is that when a muscle contracts, the cross bridges happen in sequence to maintain the contraction cycle. Take a few minutes to review the cycle with a diagram. Now there is a period of time after running a contraction cycle when the muscle cell cannot start another contraction cycle. This is the refractory period. Skeletal muscle cells have extremely short refractory periods. They will recover from a contraction cycle quickly, replenish the ATP, and move the ions back into position. However, the time it takes for cardiac muscle cells to recover, replenish the ATP, and move the ions back into position is much, much longer. Smooth also has long refractory periods. We're now going to look at these concepts in turn. A motor unit is defined as one motor neuron and all the skeletal muscle cells that it innervates. Some motor units are large, meaning one motor neuron can innervate many cells. We find large motor units in weight-bearing parts of the body. That makes sense. One action potential can make a large part of the muscle contract. Small motor units are found in muscles responsible for fine motor movement. Again, this makes sense, as one action potential would innervate a small part of the muscle and allow us to have much more control over it. A muscle twitch is defined as one stimulus and one response. A twitch allows us to see the three phases of the contraction cycle. The latent period is the period of time from the action potential and the first power strokes. While no shortening or tension is generated here, the muscle cell is actually doing quite a lot. The action potential is traveling across the sarcolemma, down the T-tubule, and causing calcium release from the Sarcoplasmic Reticulum. The calcium is binding to troponin, causing tropomyosin to move off the actin active site. The myosin is forming cross bridges. When myosin starts to power stroke, the cell will begin to generate tension. This increase in tension as we form more cross-bridge cycles is called the period of contraction. We will have a peak tension generated according to the number of action potentials that arrived. Once the action potentials stop coming, we no longer make new cross-bridges. Now, the speed at which we break the cross bridges with ATP catches up, and the filaments slide back to the original position as calcium gets pumped back into the sarcoplasmic reticulum. This period of time when the tension is released is called the period of relaxation. Not all skeletal muscle cells are the same. Some cells will go through the three phases quickly, and we can refer to them as being fast twitch. Others go through the cycle slowly, and they are slow twitch. Here we see the very slow, relatively speaking, soleus muscle. That muscle is in your calf and assists you with walking. It is a slow twitch organ. The lateral rectus muscle pulls your eye toward your ear. It's a fast twitch muscle. Fast twitch muscles go through contraction cycles quickly and also tire out quickly. Move one of your eyeballs toward your ear and leave it there for a few seconds. Your eye will start to feel odd as that muscle runs out of ATP and signals the brain that it is fatigued. However, you can walk for hours and hours, right? That's because your slow twitch cells take a very long time to fatigue. Skeletal muscles are under amazing fine-tuned control from our cerebral cortex. In fact, skeletal muscles can generate variable amounts of force. In other words, any given muscle can contract a little or a lot. It just depends on what your brain wants to do. These are some factors that affect how much force any given skeletal muscle can generate. The more motor units we fire action potentials on, the more muscle cells in a given skeletal muscle we will innervate. What's it mean? Well, the more action potentials we fire, the more cells we innervate, the more contraction we get, the more force that organ generates. Our cerebral cortex makes its best guess. Have you ever lifted something you thought would be really heavy, but then it was surprising to you when it wasn't heavy? That's because your cerebral cortex made a guess. It innervated a lot of motor units to generate a large force to lift that object. But then you didn't need that extra force because the object turned out to be light. We also do this the opposite way. Sometimes you pick up something you think will be light, only to be surprised at how heavy it actually is. Muscle cell size is a factor. The bigger the cell, the more myofibrils it has, and the bigger the force of contraction, the more cross bridges. Frequency of stimulation. This is related to summation, and we'll come back to this in a little bit, but for now, the faster you send action potentials to a muscle, the bigger the contraction force becomes. Lastly, if you stretch a muscle, first, it responds by pulling harder on the next stimulus. Skeletal muscles are not all or none in their contraction responses. Every skeletal muscle in the body can contract either a little or a lot. It is all up to the cerebral cortex. This concept is called graded muscle response. Again, you can contract a little bit of a skeletal muscle or a lot more. There are 3 graded muscle responses: wave summation, incomplete tetanus, and complete tetanus. Wave summation. Compare this to the muscle twitch. We have 2 stimulus events so close together that we don't finish the period of relaxation before starting another period of contraction. When this happens, the force of the second peak is higher than the first. That's because there was some leftover calcium still in the cytoplasm after the first action potential. We add more calcium after the second action potential and end up making more cross bridges the second time. Incomplete tetanus, the same concept, but more than 2 action potential events. We see the continuous increase in the force of contraction with each stimulus event. Again, more and more and more calcium is being left in the cytoplasm. So more and more and more cross bridges are cycling. This will reach a maximum eventually. An example is if you're holding onto something while standing in line at the grocery store. Your arms continue to hold the objects, but they are getting tired. Complete tetanus. The stimulus events are now extremely close together. There is no relaxation occurring at all. In no time at all, we hit maximum force. Unfortunately, this will cause one of 2 things to happen. 1, our brain might force the skeletal muscle to stop contraction. Or 2, our muscles might lock in a contracted state when they run out of the ATP needed to cycle the cross bridges. A sports example here is bonking or hitting the wall. Someone pushes their muscles past their adapted states and collapses or goes into muscle spasms. A smaller example of this is when you hold tension in your shoulder and back muscles for a long time, say, while listening to this lecture. Now you have the local areas of your muscles that are in tetanus and locked. You might call that a Charlie horse or a knot in the muscle. Wouldn't it be great if you could get the benefits of increasing force of contraction without going into tetanus? Well, that's exactly what happens in Treppe. Here we see multiple stimuli. However, the cells are allowed to complete a twitch before being stimulated again. We can see the increased tension generated with each peak. This time, more and more calcium is entering the cytoplasm like before. But we are also generating some heat, which is warming the muscles and making everything more efficient. Treppe occurs when you walk before you run or when weight training in repetitions per set. Now, even when you think you are relaxed, your skeletal muscles aren't. Spinal reflexes cause muscles to constantly cycle cross bridges between motor units. What's this mean? Well, even when you're lying down asleep, a few motor units are contracting in each muscle at a time, and then the motor units across the muscles are taking turns. This constant and slight contraction is muscle tone. Tone keeps blood flow going through a muscle and is a force that stimulates myofibril maintenance. When paralysis occurs, that tone is lost. We then see the muscles degenerate and weaken as that stimulus for myofibril maintenance is lost, and the cells slowly break down the myofibrils over time. Did you know that there are 3 ways muscle cells can generate tension? The method we've talked about, shortening the sarcomere to generate tension, is only one method. Isotonic. The sarcomere and muscle change length and tension is created. There are 2 ways to do it. Concentric. The sarcomere shortens. This is the method we've discussed. Eccentric. The sarcomere lengthens. Isometric. The sarcomere and muscle stay the same length while generating tension. Here we can see a real-life example of the 3. When we lift these weights, we are doing so by shortening the muscle and generating the force needed to move that load. So lifting the weight is concentric isotonic contraction. If you hold the weight up for a few moments, you are still generating tension. This is isometric contraction. Now, you slowly put the weight back down in a controlled manner. You are able to control the movement because you're still generating tension while the sarcomeres are lengthening. This is eccentric isotonic contraction. Well, now you know more than you ever thought possible about muscle cell contraction and behaviors. We now switch to metabolic processes and how they make their ATP. This is related to twitch speed. Muscle cells have 3 options for making ATP. But first, a little about ATP. This molecule is adenosine triphosphate. It is an adenosine with 3 phosphate groups attached. The 3rd phosphate group is high energy and it pretty much falls off. When it falls off, we use it to do some kind of work. For example, if we stick that phosphate to the myosin head, we can break the cross bridge. Now, when that 3rd phosphate falls off, we are left with adenosine diphosphate. ATP is so high energy that there's no way to store it, and it is used as it is made. Since we can't store energy as pure ATP, that means we have to store energy in some other way. This energy needed to make ATP is stored within the chemical bonds of other molecules. What cells will do is break Those other molecules then use that energy to make ATP. Creatine phosphate. This molecule allows for a quick recycling of ATP from ADP and another phosphate. Creatine phosphate recycling is limited and only lasts a few seconds before it is depleted. This method is used by all cells. Anaerobic metabolism, so called because oxygen is not needed. First, the cell will start with a 6-carbon glucose molecule. That is broken in half during glycolysis, and the energy is used to make some ATP. The cell then uses the 3-carbon pieces in fermentation to make lactic acid. That fermentation does not give us more ATP but allows the cell to continue running glycolysis. This method lasts a few minutes, but eventually the cells will run out of glucose. This method is used by all muscle cells but is the only option for certain fast twitch cells. Aerobic metabolism, so called because oxygen is required. First, the cell still starts with glycolysis and breaking a 6-carbon glucose into 2 3-carbon pieces. Each piece is then given to the mitochondria. The mitochondria will run the 3-carbon piece, pyruvate, in a cycle, the Krebs cycle, then a final process, oxidative phosphorylation. The end result is a ton of ATP. Now, not only can the cells use aerobic mechanisms to make a lot of ATP, they end up having options. Once the glucose runs out after a few minutes, these cells can switch to using fat supplies. Why? Because the chemistry of the Krebs cycle is very flexible. A cell just needs a carbon-based molecule. It doesn't have to be from glucose. So once that glucose runs out, the cell starts to break down fat molecules and plugs those into the Krebs cycle process. This means that these cells produce ATP for hours and hours. Obviously, this means these cells need mitochondria. And because oxygen is part of the last step, it also means these cells need lots of oxygen. If they are robbed of oxygen, they'll stop aerobic and switch to anaerobic. To quickly review, creatine phosphate recycles ATP and lasts a few seconds. Anaerobic metabolism, glycolysis and fermentation, break glucose and produces ATP, but only lasts a few minutes. Aerobic metabolism, glycolysis, Krebs cycle, oxidative phosphorylation, breaks glucose and fats and lasts for hours. Now we can overlap metabolic processes to the twitch speed. Fast glycolytic cells go through their contraction cycle quickly and rely on glycolysis and fermentation. As a result, they tire quickly. Slow oxidative are the opposite. They contract slowly but run aerobic metabolism and can contract for hours without tiring. Fast oxidative are a compromise between the two. They have a fast twitch speed but can run oxidative metabolism and contract for a long time without tiring. Now, compare the explosive and short-term action of a sprinter to the slower and long-term action of a marathon runner. Which activity is more suited to fast-twitch glycolytic oxidative cells? Which is more suited to slow-twitch oxidative cells? Humans have all cell types in their muscles. However, Not every muscle is a complete mixture of the three. Remember the lateral rectus of your eye? That's primarily fast glycolytic. However, the soleus calf muscle is mostly slowly oxidative. Within that, there are differences between human beings. Some of us have just a bit more of one versus the other in our muscles. Michael Phelps is famous, but during one Olympics, he became even more famous because he won gold medals in sprints and long swims. That's highly unusual because athletes tend to be one or the other because of the makeup of fibers within their muscles. Think about this and talk to each other about it in the A&P Cafe. And here's another example, white meat versus dark meat. White meat contains mostly fast glycolytic cell types. These muscles do not store fat because they don't use fat in metabolism. They also do not have a high blood supply because they're mostly anaerobic and don't need the extra oxygen. However, dark meat is the opposite. These cells are found in thighs and legs and are weight-bearing, so they need to contract for hours on end, which means they are slow oxidative. As a result, they store fat for use in metabolism and need an extra rich blood supply for the high oxygen demand. In addition to metabolism, aerobic and anaerobic can refer to exercise stimulus on skeletal muscles. Depending on how you stimulate the muscle, you will see different responses. Aerobic exercise like running, dancing, walking, swimming stimulates muscles to adapt for aerobic metabolism. So we generally see more blood supply, mitochondria, and myoglobin in the cell. Myoglobin allows muscle cells to move oxygen into the mitochondria. Anaerobic exercise such as weight training results in different changes. When a skeletal muscle cell contracts to move a load that is heavier than what it is adapted for, the myofibrils tear. However, the cell responds by replacing and repairing the myofilaments. With continued weight training and continued tearing, the muscle cell adapts over time by making more myofilaments. These cells also increase the glucose stored in them by making more glycogen and more mitochondria. If you exercise a muscle to the point of it running out of ATP, you force it into fatigue. Ion balances occur, lactic acid builds up, and the myofibrils are torn. The cell then goes into oxygen debt, where it replaces and repairs to return to homeostasis. This fatigue debt cycle is called the overload principle. Skeletal muscle cells always adapt to their stimulus. If you want them to continue to adapt and change, you must continue to overload them. Those who work out with weights know that after a month or so, the muscles get used to lifting that weight. In order to continue to improve on performance, the person must either increase the weight or lift more sets. Now, let's look at how different smooth muscle is from skeletal. As a reminder, cardiac uses the same sliding filament form of contraction as skeletal. It does not do extreme graded responses and is slow twitch oxidative and slow to tire in nature. Structurally, smooth muscle cells have endomysium only and often occur in thick sheets of thousands and thousands of cells. When in the walls of hollow organs, they can sometimes be found in longitudinal and circular planes. Functionally, smooth muscle can cause glands to secrete or control a tubular organ. Peristalsis is wave-like contraction to push something through a tubular organ. Segmentation is a back and forth contraction for mixing. Dilation and constriction change the diameter of a tubular organ. Lastly, some smooth muscle relaxes when stretched, but other cells may contract when stretched. Smooth muscle cells are not striated. This is due to the myofibril arrangements. Instead of being full of myofibrils perfectly stacked on each other, these cells have superficial myofibrils that are cross-hatched instead. Since the myofibrils are superficial, there's no need for deep T-tubules. So the sarcolemma have short invaginations called caveoli. Some smooth muscle cells can self-depolarize when stretched. Most smooth muscle cells in the sheets are coupled by gap junctions to ensure that they contract together in large units. Also, smooth muscle does not have terminal neuromuscular junctions. The motor neurons here have synapses that occur in chains and are called varicosities. To review, smooth muscle cells are not striated, do not have T-tubules, but caveoli, however you want to say it, instead. They do not have terminal neuromuscular junctions, but varicosities instead. They can self-depolarize. They are coupled with gap junctions. They contract in sheets. Smooth muscle cells do not use the troponin complex. Instead, release calcium binds with a calmodulin, an enzyme complex. This then activates myosin to form cross bridges and power strokes. Also, smooth muscle can grow by cell division. There are two types of smooth muscle. The type we just talked about is called single unit. You can see all the characteristics here. There is a type of smooth muscle that behaves more like skeletal muscle. That tissue, multi-unit, is found in a few places: large arteries, large airways, erector peeling muscles on our hair follicles, and inside the eye. Note how multi-unit has motor units, rare gap junctions, doesn't self-depolarize, has greater responses, and has cells that are independent. Congratulations! You've just finished Bio 2050. A very time-consuming, challenging, and stressful course. Don't forget what you've learned. You'll need it in Bio 2060, as well as pharmacy school, PT school, med school, nursing school.